protein modification, folding, sorting or targeting, packaging, transport and secretory pathway. Functions of endoplasmic reticulum. As stated above, the endoplasmic reticulum is categorized into two types, and both these types of perform specific functions. Smooth endoplasmic reticulum function. Smooth ER is responsible for the synthesis of essential lipids such as phospholipids and cholesterol. Smooth ER is also responsible for the production and secretion of steroid hormones. It is also responsible for the metabolism of carbohydrates. The smooth ER stores and releases calcium ions. These are quite important for the nervous system and muscular systems. Rough endoplasmic reticulum function. The majority of the functions of rough ER is associated with protein synthesis. The rough endoplasmic reticulum also plays a vital role in protein folding. Also ensures quality control regarding correct protein folding. The second most important function after protein synthesis and protein folding is protein sorting. Chaperones are proteins that guide proteins along the proper pathways for folding. They protect proteins when they are in the process of folding, shielding them from other proteins that might bind and hinder the process. For detailed study, please visit the website Biogenesis.com, and to remain updated, follow all the social media profiles. All the links are in the description. Ribosome biogenesis and assembly. Ribosome biogenesis is the process of making ribosomes. As ribosomes are not self-replicating particles, the synthesis of its various components such as RNAs and proteins are under genetic control. As described in Chapter 4, the expression of RPs is well synchronized at the levels of transcription and post-transcriptional modification, translation as well as post-translational modification to produce accurate stoichiometric ratio. Thus, ribosome biogenesis is a highly dynamic process of the cell articulated in multiple, highly coordinated steps beginning in the nucleolus and ending up in the cytoplasm. It involves transcription of the rRNA and RP genes, their processing, modifications, association, and proper fold. In addition to the presence of a nucleus, eukaryotic cells are distinguished from prokaryotic cells by the presence of membrane-enclosed organelles within their cytoplasm. These organelles provide discrete compartments in which specific cellular activities take place, and the resulting subdivision of the cytoplasm allows eukaryotic cells to function efficiently in spite of their large size, about 1,000 times the volume of bacteria. Because of the complex internal organization of eukaryotic cells, the sorting and targeting of proteins to their appropriate destinations are considerable tasks. The first step of protein sorting takes place while translation is still in progress. Many proteins destined for the endoplasmic reticulum, the Golgi apparatus, lysosomes, the plasma membrane, and secretion from the cell are synthesized on ribosomes that are bound to the membrane of the endoplasmic reticulum. As translation proceeds, the polypeptide chains are transported into the endoplasmic reticulum, where protein folding and processing take place. From the endoplasmic reticulum, proteins are transported in vesicles to the Golgi apparatus, where they are further processed and sorted for transport to lysosomes, the plasma membrane, or secretion from the cell. The endoplasmic reticulum, Golgi apparatus, and lysosomes are thus distinguished from other cytoplasmic organelles by the common involvement in protein processing and connection by vesicular transport. What is the sorting, packaging, and transport of proteins? Proteins are sorted into the regulated secretory pathway in the trans-Golgi network, where they are packaged into specialized secretory vesicles. These secretory vesicles, which are larger than other transport vesicles, store their contents until specific signals direct their fusion with the plasma membrane. What is the modification and packaging of proteins? The Golgi apparatus is a cellular organelle responsible for the modification and trafficking of proteins to other organelles. Proteins translated within the rough endoplasmic reticulum are transferred to the Golgi. From there they are modified and packaged into vesicles for distribution. Where are proteins modified and packaged for transport? The Golgi apparatus. The Golgi apparatus transports and modifies proteins in eukaryotic cells. How have scientists studied dynamic protein movements through the Golgi? The Golgi apparatus is the central organelle mediating protein and lipid transport within the eukaryotic cell. What is modifying, sorting and packaging of proteins for secretion? After synthesis and translocation into the endoplasmic reticulum, proteins are exported to the Golgi apparatus, a multi-compartment organelle that is the protein modifying, packaging and distribution center of the secretory pathway. The most common modification is proteolytic cleavage. Some of the pre-cleavage polypeptides are immediately cleaved, while others are stored as inactive precursors to form a pool of enzymes, or other kinds of proteins, that can be activated very quickly, on a timescale of seconds to minutes, as compared to having to go through transcription and translation, or even just translation. A major class of cleaved peptide sequences is signal peptides. Signal peptides direct the protein from the cytoplasm into a particular cellular compartment. In the case of prokaryotes, this essentially means the cell membrane, but for eukaryotes, there are specific signal peptides that can direct the protein to the nucleus, to the mitochondria, to the endoplasmic reticulum, and other intracellular organelles. The lumen plays 4 major protein processing roles: folding, refolding of the polypeptide, glycosylation of the protein, assembly of multi-subunit proteins, and packaging of proteins into vesicles. Refolding of proteins is an important process because the initial folding patterns as the polypeptide is still being translated and unfinished may not be the optimal folding pattern once the entire protein is available. This is true for H bonds and more permanent disulfide bonds as well. Glycosylation is an important modification to eukaryotic proteins because the added sugar residues are often used as molecular flags or recognition signals to other cells that come in contact with them. There are 2 types of protein glycosylation, both of which require import of the target polypeptide into the N-linked glycosylation actually begins in the endoplasmic reticulum, but O-linked glycosylation does not occur until the polypeptide has been transported into the Golgi apparatus. Vesicles, membrane-bound bubbles, essentially, pinch off from the Golgi, and other membranous organelles, carrying with them whatever soluble molecule. For detailed study please visit the website Biogenesis.com, and to remain updated follow all the social media profiles. All the links are in the description.